next here is James Marshel from Stanford University. And he's going to be talking about low threshold layer specific neural ensembles contributing to the ignition of perception.
Yeah, so we've known for quite some time that direct microstimulation of only a few neurons in cortex can give rise to remarkably specific perceptual experiences. In the case of vision, these are known as phosphenes, and it can include flashes of light, sometimes containing color, shape, or texture information, and more rarely have been reported to include even more complex percepts. This implies that circuit mechanisms exist, especially perhaps in early visual areas, that can amplify weak inputs and have profound effects on downstream areas and perception. To gain insights into this, it would be very helpful if we can train mice to report artificial percepts so that we can better understand the ignition of neural activity at the cellular level that is needed to drive specific percepts. So in terms of candidate circuit mechanisms that may underlie ignition-like effects, it has been shown in several studies using a variety of methods that within layer 2-3 of mouse V1, neurons that share orientation tuning preference are more likely to be connected, forming reciprocal connections that may act as such an amplification circuit. Whether these functionally specific subnetworks extend beyond layer 2-3 to include, for example, layer 5 cells is not known, and even more importantly, whether these circuit mechanisms may actually ignite these ignition-like effects and underlie specific visual percepts is also not known. To gain insights into these types of questions, we developed and applied a new all-optical ensemble-specific stimulation approach using calcium imaging and holographic optogenetics that allows us to stimulate specific ensembles while recording calcium activity across cortical layers while mice are viewing and discriminating visual stimuli. So to do this, we developed a new spatial light modulator holographic device and integrated 2 of these SLMs into a multiphoton microscope such that we could achieve kilohertz ensemble update rate of stimulation. We also developed a new redshifted opsin, which we called Carmine, which drives very large photocurrents on the order of several nanoamps, much larger than other redshifted opsins that we tested. And using 2-photon single-cell optogenetics, we've been able to stimulate approximately millisecond level temporal resolution of spike timing. So using a single viral vector to express both GCAMP6M and soma-targeted Carmine across large swaths of cortex, we've tested this microscope by randomly selecting dozens to over hundreds of cells to stimulate at these kilohertz rates. So here's an example where we're stimulating 25 neurons or so in each millisecond time step. We then repeat this sequence of ensemble stimulation to drive these cells at 30 hertz, and then record their calcium activity, confirming that the great majority of these cells actually respond to the stimulation. So we've extended this approach to stimulate neurons across layer 2-3 and layer 5. Here, the green circles indicate the cells that we're stimulating, and you can see that we can drive robust responses across the volume. So in our first experiment, we started with a naive cohort of mice that were not performing any specific task. We recorded calcium signals while the mice were viewing these drifting gratings and found cells that were selective for one or the other orthogonal grating. These cells were scattered across the volume, and for controls, we also designate 2 random ensembles that will have the same number of cells as the tuned ensembles. So we can stimulate these specific ensembles with the holographic approach and confirm that we can drive robust responses in these cells. And then we're going to perform a series of analyses where we ask whether tuned ensemble stimulation can evoke similar dynamics in the surrounding population that resemble visually evoked dynamics. Importantly, these unstimulated cells that we'll use for these analyses, of course, exclude all stimulated neurons, but also any neurons that are nearby those cells and any neurons that are directly above or below those cells at any depth. We use sparse logistic regression to decode the visual stimulus based on the unstimulated population responses to visual-only trials. Ranking the neurons based on their weight in the model, we can see that the analysis pulls out neurons that with selective responses for either the 0-degree or 90-degree visual stimulus, with the difference between these responses shown at the bottom. If we take the same classifier without any additional training and apply it to the tuned and random ensemble stimulation conditions, we'll see whether we'll be able to discriminate the condition based on the unstimulated population responses. So first, if we look at the responses under these different conditions, again, using the classifier that was trained on visual-only trials, during the tuned ensemble stimulation conditions, we see that similar highly weighted cells show responses that again show this differential response, but we don't see this during the random ensemble stimulation cases. So indeed, the classifier can perform well at discriminating the visual stimuli, can also discriminate the tuned ensemble stimulation, but does not discriminate the random ensemble stimulation conditions. And if we use principal components analysis to gain greater insights into the neural trajectories over the course of these different trial types, we can see that on visual trials, not surprisingly, there are largely divergent activity patterns given the orthogonal visual stimuli that we're showing to the animal. If we keep these principal components constant to then visualize the other conditions, we can see that during the tuned ensemble stimulation, we evoke a very similar pattern of dynamics resembling the visual-only trials, but this is not seen during random ensemble stimulation. And so given that we can generate these very visual-like looking dynamics, we're excited to ask whether the mice were actually potentially seeing a stimulus resembling the stimuli we were trying to emulate. So to gain access to this type of question, we trained water-restricted mice to lick to vertical gratings and withhold licking to horizontal gratings. They can do this task to very high performance after training over several sessions. We then add in lower contrast visual stimuli to map the perceptual threshold below which the animals can no longer do the discrimination. We then put the mice back into the behavior, and now on a subset of trials, we'll present low contrast visual stimuli and also tuned ensembles or size-matched random ensembles to see their effects on the behavior. So if we look at the animal's behavior going across their perceptual threshold, we can see that specifically when we stimulate the tuned ensembles in combination with these low contrast stimuli, the animal's behavior modestly improves. And we do not see this during random ensemble stimulation. Even more strikingly, after the animals had performed this contrast modulation ramp experiment, they were able to discriminate the tuned ensembles without a visual stimulus present. This was in contrast to their behavior before the contrast ramp experiment, in which really only one mouse was able to do the tuned ensemble discrimination without visual stimuli. So given these apparent learning effects, we looked at the behavior during our random ensemble stimulation cases, and I'll just mention that the same random ensembles were used in all trials and across all sessions, basically held constant for all behavioral sessions. So the results of these data were much more variable, but clearly some of the mice had learned to discriminate the random ensembles without a visual stimulus present. So to try to tease apart the different effects that may be going on in these behavior experiments, we moved to an entirely new field of view and found a non-overlapping volume of cells, defining a new set of tuned ensembles as well as a new set of random ensembles. We immediately put the mice back into the behavior, and they were easily able to discriminate the tuned ensembles, but could not discriminate these new random ensembles. And this indicates to us that the tuned ensemble stimulation provides a more immediately interpretable signal to the animal, perhaps resembling to some extent the visual stimuli that we're trying to emulate. So we went back to these same ensembles day after day for several weeks. The animal's behavior doing this tuned ensemble discrimination task was highly reliable and robust. The animal's performance was an average of 90% correct, and this was on par with their behavior on visual-only trials that were randomly interleaved on the same sessions. We repeated our logistic regression analysis, finding largely consistent results compared to the untrained cohort. The classifier and the animal's behavior was very high for the visual trials as well as these tuned ensemble stimulation cases, and now also could somewhat discriminate the random ensembles, indicating likely local network plasticity, linking these random ensembles to the same tuned subnetworks. Looking at the PCA across all of these sessions, each of these traces is the average neural trajectory from one of these sessions. We can see that these dynamics are highly reproducible over time, and that the tuned ensemble stimulation largely recapitulates the visually evoked dynamics. Interestingly, one of the mice that had actually learned to do the random ensemble discrimination, we see actually a similar pattern beginning to emerge. So we finally turned to studying the impact of stimulation of neurons in either layer 2-3 or layer 5. Stimulating only layer 2-3 cells led to selective recruitment of similarly tuned neurons within layer 2-3, as well as across layers, recruiting isotuned neurons in layer 5. We also saw strong selective recruitment of similarly tuned neurons within layer 5, but interestingly, did not see much recruitment at all of layer 2-3 activity, and we saw no evidence that there was any specificity in this recruitment of layer 2-3 activity. We systematically dwindled the number of neurons in these experiments as we stimulated neurons in each layer, and we found that there was a critical number of neurons that the animals needed in order to perform the task, and that was right around 20 or so cells, after which their performance rapidly dropped off. We saw a very consistent pattern when we looked at the performance of our classifier, and if we compared layer 2-3 stimulation to layer 5 stimulation, we generally found that layer 5 stimulation was more potent at driving these ignition-like effects and driving the behavior of the animal, likely reflecting the important projections of layer 5 to other cortical and subcortical areas. We finally developed a simple neural network model to try to recapitulate these ignition-like effects. These models could produce similar effects with comparable numbers of stimulated neurons out of a population of order 1,000 cells, but importantly did not show spontaneous ignition events when taking into account the low spontaneous firing rates that are generally observed in mouse visual cortex. In this way, V1 may be operating near a point of instability that would be rarely crossed without input to the system. So I definitely want to thank the many people who are involved in this study, in particular Tim Machado, Sean Kieran, and Yoon Kim, as well as Surya Ganguly and Carl Deiseroth, and of course all the funding sources.
Just wonder so first of all, the PCA that you did was in the coordinates from the tuned, and then you looked at random?
Thanks for asking that question. Actually, it was based on the visual-only trials. So basically, all the analyses followed a principle where we defined the classifier, we defined the PCA based on visual-only trials, and then projected the data from the different ensemble stimulation conditions onto those same axes.
And did you try to take an ensemble of let's say, untuned, but neurons that have high noise correlation? So maybe they do the same thing, but just not visual, and see if that can be learned?
Yeah, so we literally chose the cells at random from the surrounding population. I think there's a lot of really interesting things to follow up on other types of cells that we could choose, but the tuned ensembles were highly tuned for the visual stimuli.
Did you think of trying natural images instead of tuned gratings?
Yeah, I would love to do that in the future. Yes, of course. We started with a very simple stimulus, easy to find the cells rapidly. These experiments are sort of a closed loop type of experiment where we need to find tuned neurons in a short amount of time, so it's a good place to start. But yeah, absolutely.
Okay, thanks, James.